Hey everybody, my name is Wes Anderson. I'm a Ph.D. candidate in the Department of Wildlife Ecology and Conservation at UF. I'm advised by Dr. Raoul Boughton, who's based here at the Range Cattle REC. And before I get started, I just want to tell the people logging in through the webinar that you can type your questions whenever you want. I'll get to them at the end. All right, so today I'm going to discuss the research that I've been doing for my dissertation. It looks at the impacts of wild pigs on wetlands and aquatic communities across the Florida rangeland. All right, so where does wildlife conservation and management occur? When people think about that, maybe traditionally they think about Africa. This picture was taken in Botswana of some elephants. Or maybe more regionally or at the state level, we think about the Everglades. And then when we think about wildlife management, we're usually thinking about game species, whether it be deer, bobwhite, waterfowl, things like that. That doesn't have to be the case. So almost 40% of the entire world's land area is under some form of agriculture right now. And over a quarter of that is rangeland, whether it be cattle, goats, sheep, things like that. Now in Florida, rangeland accounts for 12 million acres, or approximately a third of our whole land area here. So they're predicting that Florida's population is going to double by 2060. During that doubling, a lot's going to be converted to urban areas, and we're projected to lose 3 million acres of agriculture. So a lot of the land is covered in agricultural systems, and we're also losing those agricultural systems. So wildlife conservation and management in agricultural lands, like rangelands in Florida, is critical to conserve biodiversity. So this talk is going to focus a lot on amphibians. Why amphibians? I guess first off, because I like them. I grew up catching salamanders and frogs. Realized that I could continue doing that for the rest of my life. So frogs, there are a few reasons why they're important. They're an important prey item. Everything loves to eat frogs, whether it be birds, mammals, other frogs, fish. They also consume a lot of insects. So I don't know, I'm thankful for the tree frogs on the windows because I know a lot more bugs would be getting into my house if they weren't there, eating a bunch of them. And maybe most interestingly, they're important for energy flow from the wetlands or the aquatic environments where they live up into upland habitats. So the tadpoles live in the water, but then they metamorphose, and a lot of those species will actually go up into the uplands. So that energy is getting transported out of the water. So let's talk about salamanders, two types in particular that live in Florida that a lot of people don't know about. Those include sirens, which are these guys right here. They're one of the biggest salamanders in the world. They can get up to 3 foot long. They've only got 2 front legs. And what's unique about them is if their wetland dries out, they just dig down into the ground. They're going to slough off some of their skin and make a cocoon, wait for the water to come back. And that leaves them vulnerable to whatever's going to eat them, which I'll talk about later. They're also amphibians. They get even bigger. They can be almost 4 foot long. If you can see, it has 2 tiny forelegs. They actually have 4 legs, but only 2 toes, and their limbs are really reduced. Now, amphibians in general are experiencing declines throughout the entire world. It's a real biodiversity problem, and they've been called canaries in the coal mine because they're pretty sensitive to environmental change, at least some are. So if we can evaluate the status of our amphibians, we can get a better idea as to just the general health of the environment. So whenever you're trying to conserve biodiversity, one thing that you have to worry about are invasive species. In Florida, the wild pig is an important invasive species. It's invasive because it causes environmental impacts, it causes economic impacts, and also human health concerns. And I know that probably everybody here has seen hog rooting before, but I don't know if you've seen it in action. So here's just a video stitched together from our game cameras over at Buck Island Ranch. And so this occurred over the course of a few nights. These are still photos stitched together. But you can see that just 2 pigs can root up a pretty big area in just several minutes at a time. All right, so I'm going to discuss 4 things during this talk. 1, we're going to examine wild pig diet. I'm going to discuss especially how that relates to amphibians. Then we're going to talk about some of the research we've done with drones and trying to quantify rooting. Looking at impacts of salamanders. This is my pet siren right here. I actually caught him at Ona back in 2015. He's doing well. And then impacts on tadpoles. And so when we discuss impacts, there are two different kinds that there can be. It can be a direct impact by the pigs, which they're consuming the animals, or it can be an indirect impact. So with all the rooting that pigs do, they really like to target wetlands, especially during the winter when they're dry. So they're causing a lot of habitat disturbance. And so that would be an indirect impact. All right, before I get started more with the study, I'm going to just talk about the study site a little bit. Buck Island Ranch, has anybody here been there besides Crawler? So it's over in Highlands County. It's over 10,000 acres in size, and it's a full-scale commercial operation. They run about 3,000 head of cattle there. Across the landscape are a ton of wetlands, the majority of them seasonal, over 600. It's also heavily modified. There are hundreds of miles of ditches, which the blue that just showed up is all the ditching throughout the site. And so for my study, and most of the things I'll be talking about, I'm focusing on 36 wetlands between about an acre and 3 acres in size. And you can see them, they're all in green right there. So they're scattered throughout the site. They're split between improved pasture, which is predominantly Bahia grass and is usually ditched more, and then semi-native pasture, which is it has a mosaic of Bahia grass and native plants. All right, so starting with wild pig diet. So we collected a lot of pig poop. We did not actually sift through that poop. We sent it off to a lab in Fort Collins where they did DNA analysis on it. And so using a technique called DNA metabarcoding, where for plants we're targeting one marker, for animals and fungi we're targeting another, and then we're able to generate these operational taxonomic units, or OTUs. So actually, let me talk about the methodology just a little bit more. So this occurred over a whole year's time. The year was broken up into 6 2-month blocks, and the ranch was also divided into 5 different areas. We wanted to do that just so we weren't collecting the fecal samples just in one area. Every 2 months, we collected at least 5 fecal samples from each area. That's my fiance, Christy. She's helped me a lot over the past 3 years with pretty much all my field work. She'd drive down every weekend, so she'll show up more. So once we sent the fecal samples off for analysis, what we got back was this list of OTUs, or operational taxonomic units. So what it's doing is each sequence that it can parse out, it's then identifying or relating that to a sequence in an online database. So we have here the top line, that would be oaks. We just have a whole list for both plants and animals. But then what we had to do is look at what occurred, what was listed, and then do kind of quality control to say, well, actually this doesn't occur at the ranch where we're working. So what could it be? So if we could then get it down to at least the family level, we kept it. If we couldn't, then we discarded it. So I'm going to focus on wetlands and amphibians. So this graph that I'm showing right now, it's starting at the beginning of our collection period, which is in March 2016, ending in February 2017. And what this is showing is the percent of the diet of animals that are wetland dependent. So from March till October, you're not really seeing anything there. They're consuming very few animals that are wetland dependent. But in November, it ticks up to about 10% of the diet. By January, February, it's about 20%. So out of all the animals that they consumed, there were 3 amphibian taxa. One was the eastern narrow-mouthed toad. Oh, that's not great. One was the eastern narrow-mouthed toad. They're a fossorial species, so that means that they spend most of their time underground. So it's not really all that surprising that the pigs were digging them up. When I was making this garden just, I don't know, a few hundred yards away, I was digging them up myself. More interesting to me is that they were consuming salamanders, and just during January, February. So we got sirens, which I showed you a picture of, and then these little guys are dwarf sirens. They don't get nearly as big, usually around 6 inches or so. And so this was only from 6 fecal samples. Let me move something around real quick. This was only from 6 fecal samples, but it was concentrated in just 2 months. So if we think about that a little bit more, that was 6 out of 31, which is about 19%. So if a pig's using the bathroom once a day, and there are about 500 pigs on the ranch, over say a 3-month period during the winter, they could be consuming 8,000 or 9,000 of these salamanders, which could potentially have an impact on their populations. 1 second. So I'm going to move on to the next topic now, which is using drones to quantify rooting, which I know some of you that have been here before have heard a little bit about. But so we want to be able to develop a protocol that allows us to standardize how we're collecting these data. We're flying over those study wetlands that I showed you, the 36. We're going to take those images that we're taking with the drone, mosaic them into a single georeferenced image. And then we're going to perform spatial analyses on them so we can delineate and then quantify how much pig rooting there is. So pretty much the process is going from a drone all the way to this georeferenced image that has all the rooting drawn out. We're also going to, I'm going to touch briefly on the impacts of swine removal or pig removal on wetland damage across the dry season. So the drone that we're using, it's a very popular drone. It's a DJI Phantom 4. And when we have the drone out in the field and I'm near my 36 wetlands, then I can program a mission. I'll specify all the different attributes I want for the mission. So say the altitude I want it to fly at, the speed, how much overlap I need between pictures. And then I just let it go. And we're going to do that 4 times over each dry season. So starting in November, December, ending in May or June. So each wetland, depending on the size, it'll be 35 to 140 images. And this is just sequential images that the drone is taking while it's flying. And so what I'm showing now is a mosaic. So that's based off of a decent amount of images, I think around 60 or 70 that have been stitched together, and you can see the boundary of the wetland there. And what I just blew up there is hog rooting. So it's pretty readily noticeable on a lot of these mosaics, but it would be very difficult to do it manually, maybe almost impossible. It would take a long time. So we've developed a way to do it much quicker and more accurately. So what we first need to do, once I've combined all these images from a flight together, is classify the raster. So each pixel that you see is made up of 3 different color bands. So there's a red band, a green band, and a blue band. What I'm getting ArcGIS to do is I'm training it to identify what the difference is between a rooted area color band and a non-rooted area. So in orange right there, which is a little hard to see, those are in rooted areas. The blue is in non-rooted. So now I've trained the program to identify at least a subset of the pixels, what is rooted, what is non-rooted. Then I'm going to run a maximum likelihood classification analysis where it's going to take every pixel in the mosaic and it's going to classify it as either rooted, not rooted, or in some cases it can't classify them. We run into that especially for trees and things like that. So there are a few steps then once we run our maximum likelihood classification where we're just aggregating the polygons, removing some of the smaller ones, and then once we get to the final step, we've clipped it to the boundary of our wetland, but this program isn't perfect. So it's going to identify a lot of the areas, or it should identify all the areas that are rooted, but then it's going to identify areas that aren't rooted as well. So then we need to go through grid cell by grid cell and pick out, well, is it rooted or not? If it's not rooted, then we're going to delete it. So you can see here, this is correctly classified. This is a rooted area. This is incorrectly classified. So for whatever reason, it picked out the tops of this vegetation right here. So I would delete that out. Once that's gone, I've deleted everything. We can calculate the amount that's rooted within the wetland. So in this case, a little over 2% of the wetland is rooted. All right, so I'm going to discuss a little bit now the impacts of rooting after pig removal. So in 2016, in fall of 2016, we removed pigs south of the Harney Pond Canal, which is that area highlighted in, surrounded in white right there. We removed about 100 pigs. So what we want to know is how quickly will the pigs recolonize? Can we, is there a marked difference in the amount of rooting between the north and the south? So this is showing data from all 11 wetlands in the south and then 10 wetlands in the north. The wetlands in the north are split between improved pasture and semi-native pasture. So in the south, only 2 of the wetlands had rooting. Here's some no rooting, some very limited rooting, only 2%. Whereas in the north, 9 out of the 10 had rooting. It's really a little over 2% there. Got over a quarter of the wetland rooted in that flight, or in that wetland, and then over 40% rooted there. So it does vary between areas, but what we're seeing is a marked difference between the wetlands in the south and the wetlands in the north. So just some summary statistics. On average, the improved wetlands in the north had 16% rooted. Semi-native in the north had almost 5%, whereas in the south, the semi-native wetlands had less than 1% rooted on average. And this graph is just showing that, so once I ran a post hoc test, I was able to determine that, yes, there's no difference between the amount of rooting and improved pastures in the north or the semi-native pastures in the north, but there's a difference with both from the semi-native pastures in the south. All right. So we're going to talk about salamanders a little bit now. They're probably my favorite. So does rooting in wetlands impact aquatic salamanders? So it could impact them two ways. We know from the diet study that the pigs are getting into the wetlands during the winter, and they're actually consuming these salamanders, specifically sirens, which are burrowed down, estivating underneath the soil. So they could be getting in, rooting, and directly impacting the salamanders, or just by changing the habitat through the rooting. Maybe they're not specifically targeting salamanders, but if they're rooting a lot in these wetlands, maybe just the habitat disturbance could impact them as well. So I've done trapping over the course of 3 summers, from July until November, and the conditions have always varied. It's very difficult to work in wetlands, if any of y'all have, because you never know, well, are they going to be wet this year? Or are they going to start drying up? Or maybe a hurricane's going to come through and then I can't get back on the ranch for 2 weeks. But so each wetland, I had 15 crayfish traps. There's Christy again, next to one of the crayfish traps. So they're about almost 5 foot high. They've got 1/2-inch mesh, and they've got 3 funnels at the bottom. So I'm not baiting them or anything. I didn't really want to, because I don't know how the different species are going to react to it, I didn't want to influence them in any way. So they're just wandering through and getting trapped, waiting for me to check them the next day. So 15 crayfish traps over 5 days for each wetland. Any salamanders that I captured, I collected them, brought them back to the lab where I measured and marked them and released them. It's very difficult to measure a siren. They're very slippery. The best thing that I can do is try to make a little salamander burrito, roll them up in a wet towel. I can get a tail length on them. But then I take a picture of them next to an object of a known size, in this case a ruler, and then using image software, I can calculate actually how long their total length is, subtract out the tail. So over the course of 3 years, I caught 84 greater sirens, 8 lesser sirens, which they're just a little bit different in color. And then you might have guessed by their name, they don't get as big as greater sirens. And then 16 two-toed Amphibians. Now a lot of these pictures, you'll see me with the sirens in my hand. The Amphibians, I don't ever put them in my hand because they bite. And I've seen pictures of people that have needed stitches after being bit by them, so trying to avoid that. So with these 3 different species, I was seeing different distributions across the ranch. The greater sirens, they seem to occur everywhere. There were very few wetlands that I didn't capture them in, and there didn't seem to be much of a pattern. Whereas with the lesser sirens, I only caught them in 4 wetlands, and all 4 of those wetlands were down in the south. So those are all semi-native pasture. There's a chance that they can't deal with the ditching very well, or maybe there are fewer fish, which I should be able to answer later. And then the two-toed Amphibians, they followed a same pattern or a similar pattern to the lesser sirens, but I did catch them in some improved pasture wetlands as well. So that one's a little bit harder to interpret, and I need to do some more analysis before I can really tell what's impacting two-toed Amphibian distribution on the range. So when I'm checking these traps, I'm not just recording the salamanders. I'm only collecting the salamanders, but I'm recording everything that I'm capturing in these traps. So after 3 years, I've checked traps 7,500 times, or a little bit more so. So I've generated a lot of data on what's living in these wetlands. I've recorded fish, snakes, turtles, invertebrates. So I'm just going to touch on the other species that I've captured and just highlight some of the patterns that I'm seeing, maybe give a little bit of just natural history data. So the most common thing that I was capturing in a crayfish trap turned out to be a crayfish, which they work well, not surprising. This was the only thing that I actually didn't identify the species, because we have several species there, and it would take the number that I was catching, it would take a very long time to try to identify each one. So that was all just recorded as crayfish. This is an African jewelfish. As you might have guessed by the name, it is not native to Florida. It's native to sub-Saharan Africa. They're a popular aquarium fish, and that's how they got to Florida. Somebody didn't want it anymore, dumped it into a canal, and they've spread through the southern half of the state. They get about 6 inches long. They're hyper aggressive. They're a type of cichlid. So they'll run off all the fish. They'll eat lots of different things. Presumably tadpoles. I'm sure that they do a number on tadpoles. But this was the most common fish that I caught the entire time. I caught thousands of these. Also, another non-native. This is a brown hoplo. It's a type of catfish from South America. I suspect that their impacts aren't nearly as great as the African jewelfish, but they were also very common. Getting into some native stuff. These are striped mud turtles. They're one of the most common turtles at the ranch. They occurred throughout the ranch, and just a cool little anecdote relating this to ranching is people have recorded them foraging for bugs around cow pies. That's pretty cool. And they're the second smallest turtle in the United States. They're really cool. They're really cute. So whereas I found the striped mud turtle throughout, improved wetlands, semi-native wetlands all across the ranch, the eastern mud turtle, its cousin, I only ever found in semi-native wetlands. So I would find them in the east part of the ranch, the west part of the ranch, the south part of the ranch, but never in the center where the improved pasture was. This is one of my favorite snakes. I've caught very few of them. I only caught 2 in my traps over 3 years. But this is called an eastern mud snake. They actually get really long. They can be over 6 foot long, and they specialize on eating amphibians and sirens. So I don't know, maybe my amphibian numbers were too low, or the amphibian population's too low at the ranch for them to really be common, or just they're not very susceptible to getting captured in crayfish traps. But that was one I caught this year. Finally, the island apple snail. So this is another non-native species. We do have a native species of apple snail in Florida. This is not it. The native one is more tied to permanent bodies of water, where this one can invade wetlands or more temporary bodies, and it'll just estivate in the ground, kind of like the sirens do, to overwinter. But I have been seeing some differences in the distribution of these guys, or the abundance of these guys across years. So I think that there's something going on. There are a lot of limpkins there now. Maybe the limpkins finally have them under control, sort of, but I need to do more data analysis to figure that out. All right, finally getting to impacts on tadpoles. So the pigs aren't really, or I doubt that they're preying much on the tadpoles. That's because the pigs are mostly rooting the wetlands during the winter. The tadpoles are, so they're rooting when the wetlands are dry. The tadpoles are in there after the wetlands fill up during the summer. But there's a lot of habitat disturbance. So you can see this is the edge of one of my wetlands. All the dark brown there is a recently rooted area. So is that rooting or habitat disturbance affecting tadpole growth, survival, and species richness? So I dip netted all my wetlands for tadpoles from 2016 to 2018. A dip net is pretty much just like a butterfly net, but for the water. For each dip, I would record which species I was catching, how many, and the developmental stage. So did it have back legs yet? Did it have front legs yet? Was its tail almost gone? So for non-rooted wetlands, I would only do 25 dips in there. If it was a rooted wetland, I'd do 50 dips, half in the non-rooted, half in the rooted. And I was only dipping in 4 to 16 inches of water. I found if I went any deeper than that, then I really couldn't tell if I was catching fewer tadpoles because there were just fewer there, or was it because I'm not effectively sweeping through the water column that well and they're just able to get away. So I'm just going to summarize a little bit of information from this past field season, which this was the best field season. There are no hurricanes. There wasn't a drought. I was able to spend a lot of time in the wetlands. So this past summer, I captured almost 2,000 tadpoles in these wetlands. 14 of the 36 wetlands had some rooting in them. It varied. But this graph, so I sampled each wetland 3 different times. There was about 2 weeks between each sampling period. So N1 or NR1, R1, that's the first sampling period. You can see that there's a marked difference between the rooted, which is the dark blue, and the non-rooted, which is the light blue. So in the first sampling period, there are almost 30 tadpoles captured across all species on average, whereas in the rooted, there were fewer than 10. And that pattern continues across time. So the second survey, there's still a difference between the amount of tadpoles in non-rooted and rooted, and similarly with the third survey. Why there are fewer tadpoles across time is because some species just breed at the beginning of the summer. You know, they're done early by the end of late June, and so I'm just capturing fewer species over that time. So just some other patterns that I'm seeing with the tadpoles, because not all frogs are created equal. They're using this environment differently. Squirrel tree frogs. So this one is getting close to metamorphosis here. It's got its front legs, its back legs. Still got a long tail though. But squirrel tree frogs were the most commonly captured species at the ranch, at Buck Island Ranch. They would occur in both improved pasture wetlands, semi-native pasture wetlands, and I caught almost 1,000 of them this past year. They're also pine woods tree frogs. So as you can see, they've got really red tails. That's to divert the predator's attention away from the body, and so hopefully they can get away. But pine woods tree frogs were only found in semi-native pastures. Similarly, the barking tree frog was only found in semi-native pastures. There are actually only 3 wetlands that I was able to find this in. And in the past year, I only caught 7 of them. Compare that to the almost 1,000 squirrel tree frogs that I caught. Finally, southern cricket frogs. So southern cricket frogs are one of the only reliable things that you're going to catch throughout the entire summer. They're probably still breeding in the wetlands right now. But there weren't too many differences as far as between improved pasture wetlands and semi-native pasture wetlands. I just wanted to highlight this species because they're contributing a lot to the number of tadpoles I'm catching during the second survey and the third survey. So there's a lot left to be done. I've also collected water quality data and vegetation data, or in this case, Christy again is helping me collect it. And those variables are going to be incorporated into future analyses. All right, so just to summarize things that we've talked about so far. So with pig diet, the pigs at Buck Island Ranch, they're definitely eating amphibians, and they're consuming salamanders in the winter, which is important because, I mean, a lot of my research hinges on, well, what impacts are pigs having? And we determined this impact, which nobody's really looked at before. And it provides further support as to why, you know, pigs like rooting in wetlands so much during the dry season. So drones and rooting. So we developed an efficient method to measure this rooting. I showed you just a little case study as how we can use this methodology to then look at what are our removal efforts having on the amount of rooting between different areas on the ranch. And finally, so for the rooting data over the past 2 years, I'm gonna be summarizing all that. I'm gonna do the analyses on all the different wetlands, and I'm gonna use that as variables to help look at how rooting is impacting both tadpoles and salamanders. So impacts on salamanders. So I neglected to mention that, so I did mention that I marked them. I marked them with a pit tag, which if you've got a dog or cat that's microchipped, it's the exact same thing. So I marked them with a pit tag. I didn't recapture a single one during the entire time. So I cannot estimate population size, but what I can do, especially with the greater sirens, which, you know, I had 84 captures of them, I can examine factors that impact both their occupancy. So what factors cause them to be in a wetland, or what influences their presence in the wetland, but then also detectability. So, you know, even if the siren is there, am I able to detect it? And I think especially for detectability, I need to look at some weather data, because for example, in a wetland, maybe I trap 1, 2, 3 nights, I wouldn't get any sirens. The 4th night, I would get 3 the next day. So there's probably something weather related, I would assume, that is impacting their movements and influencing their detectability. So impacts on tadpoles. What I've shown right here is that at the broad scale, when we lump all the tadpole species together, there is a significant effect within a wetland. So if a wetland is rooted, there's going to be more tadpoles in the non-rooted areas compared to the rooted. And that doesn't necessarily mean that the frogs are laying fewer eggs, but it probably means that not as many tadpoles are surviving and reaching metamorphosis. So, you know, if 50% of the wetland is rooted, then that 50% is not really suitable habitat anymore. And any tadpoles that are there will have a much lower chance of making it. But there are also landscape effects. where the wetland's located, what pasture type it's in. Those things, those things I still need to determine. A lot of analyses left. All right, so before I wrap up, I just want to acknowledge a few groups for funding USDA APHIS Wildlife Services, which supports a lot of our wild pig work, Prairie Research Incorporated, which who I've gotten a small grant through, and also the Society for Wetland Scientists South Atlantic chapter. I've got a small grant from them to support my research. I also want to acknowledge MacArthur Agroecology Research Center, which is based at Buck Island Ranch and is a division of Archbold Biological Station. They've helped me out a lot. They let me do my research there. Been living on site for the past 2.5 years, and I really appreciate the opportunity to do some cool research there. Finally, I'll mention some people who have helped me. Christy Larkin, my fiance, been with me in the field, coming down a lot of weekends over the past 3 years. Tyler Buckley, who has really helped this year with helping me get the crayfish traps out, bring them back in. And also Noah Davidson, who helped me with the dip netting for the tadpoles, and also collecting some environmental data. So with that, thank you. And if you have any more questions that I don't get to answer right now, please contact me at this email address. Thanks. All right, do we have any questions? Yeah. Mm-hmm. That's more of a question for Dr. Boughton. And we may still be analyzing those data. There are 100 taken. Not in the south, I do not. And with that ranch, we got the Harney Pond Canal on 3 sides of it in the north, and it's just on 1 side in the south. So we've also done removal in the north, but the ability to recolonize is probably impacted by the Harney Pond Canal. Any other questions? Oh. Sure. So just 1 second. So the last question I answered, or Dr. Boughton also answered, was how many pigs were originally there in the south before they were removed, and which we estimated right around that size, and how quickly did they recolonize, which we have not figured out yet. Yes. Mm-hmm. Yes. Yeah. So each picture, it has a GPS tag in it. So when the drone's taking the pictures, each picture has a GPS tag. But then we use an online service to say, take the 80 pictures or so, we upload all of them, and then it'll stitch them together into a complete picture for us. Mm-hmm. Yes. So it shouldn't. That's a good question though. I mean, so we're not analyzing the pixel colors from the original imagery. We're then analyzing whatever is spit back out. So it's pretty remarkable with the drone. Like even cloud cover will impact the color so much. So I don't think that. So in a single flight, there will be a lot of variation even within the rooted areas. So I don't think them being stitched together is really negatively impacting. Okay. I don't think I repeated that, which I should have. So it was asked if we're stitching the pictures together to create the mosaics, does that impact our ability to then analyze the rooting, which I don't think it does because there's already a lot of variability. All right, so I'm going to get to an online question. Hey Wes, what do you think the rooting impacts What do you think rooting impacts the tadpoles? Do you think it is due to water quality changes or vegetation structure? So that's something that I'm going to be looking at, incorporating water quality and vegetation. So tadpoles have a very high mortality rate. Most of them are going to get eaten before they can turn into frogs. So I really think that vegetation structure has a lot to do with it. They don't have anywhere to hide. There's an open area that's been rooted, really disturbed by pigs. So they're much more susceptible to predators, like those African jewelfish that I was talking about. Any more questions here? Should I answer some more online? All right, next one from online. Can your data be extrapolated to hogs having a similar effect statewide? Well, some of it can, surely. And so the sirens, the Amphibians, they have, they're throughout the southeast. There are actually other more, so those two species aren't really imperiled at all. We do have others in North Florida. The one-toed Amphibian, it has a very restricted range. And at least part of this study shows that the hogs are actually getting in. They're rooting up estivating salamanders. So it would be worthwhile to look at it up there, because the same thing's probably happening up in the Panhandle too, with the different species. Any more questions? Yeah. Okay. So the question was, how was it determined what part of the pig's diet was amphibians? So what we did when we analyzed the fecal samples, we got that whole list of operational taxonomic units. So each one was related to a different species or a different genus. And also included in those data were the number of reads. So how many times was it able to be sequenced? And that's a good proxy for the amount that was in the fecal sample. So what I was presenting here, it was just the occurrence of the, if we detected the DNA at all from frogs or salamanders, that's all that I was presenting here. But we can compare, the amount somewhat. There are some caveats to that. But so yeah, we just had the 6 samples with salamander DNA and the few with the narrow-mouthed toad. And there could be, from a single sample, there's going to be a lot of plant species for sure, and then there's usually several animal species as well. So yeah, each sample will have a whole bunch of different species associated with it. Did that answer your question? Or? Yep. All right. Well, if there are no more questions, thank you again. I appreciate it.